In this video, we will discuss about how this KRAS suppress the FAS expression in the pancreatic cancer cells and whether this overexpression of these FAS receptors helps in the apoptotic of the cancer cells with the detection by immune system or not, that we will discuss in this video. So let us start with the panel A, where they have described what kind of study they did in this experiment. So first, in order to check which kind of T cells usually helps in interacting with this FAS receptor that are expressed on the pancreatic cancer cells. So based on the previous experiments, they find that there is overexpression of CD4 plus T cells and CD8 plus T cells. So these are the two candidates that they used to check whether these two plays a role towards triggering apoptosis in the tumor cells once they interact with the FAS receptors. So in order to check that, they use this anti-CD4 and anti-CD8 T cells to deplete those T cells and check whether there is a decrease in the whether there is tumor regression happens or not. So first, what they did is they take the mice model and orthopedically inject these KRAS mutations, which is doxycycline driven, and after some times when the KRAS mutations are on, they introduce this anti-CD4 or anti-CD8 antibodies. After 10 days, they turn off the KRAS, and out of 5 days of these anti-CD4 or anti-CD8 antibodies injections, they turn off this also. And what they have find out is that we will discuss what they have find out, but this is the study that they did. So first they check whether this anti-CD4 anti-CD8 works or not. So when they use, this is the control experiment where we can see that anti-CD4 and anti-CD8 cells, and when they use this anti-CD4, we see the no anti-CD4 cells, indicates that this anti-CD4 works towards depleting CD4 T plus T cells. And similarly, for when they use anti-CD8, they do not see any CD8 plus T cells. And these things are further quantified in the panel C, where we can see that in the case where anti-CD4 anti-CD4 T cells used, they do not see any CD4 plus T cells, and when they use anti-CD8 T cells, they do not see any CD8 plus T cells. And when they check this effect of this anti-CD4 and CD8 plus T cells on tumor weights, what they have find out is that when they use this anti-CD8 T cells, they do not see any difference in the tumor pancreas weight compared to the control. Whereas when they use anti-CD4 T cells, they see a decrease in the tumor pancreas weight compared to the control. Indicate that it could be CD8 plus T cells that is interacting with the FAS receptors on the tumor cells. And similar results they find out from the survival experiment, where they can see that where they use anti-CD4 antibodies, they see the tumor survival rates are high, whereas when they use anti-CD8 plus antibodies, they see the tumor survival rates are low. So this further substantiates their previous finding that CD4 plus the CD8 plus T cells plays an important role, and it might be interacting with the FAS receptors on the tumors. And thereafter they check the H&E staining of the in vivo mice, and in this case also they find out that when they use anti CD4 plus T cells, they see that more tumor regressions, whereas anti CD8 plus T cells with KRAS mutations on and off, they do not see any much tumor regressions compared to the control. And these things they have quantified in terms of the different areas of the pancreas, where they can see that when the KRAS mutations are on, most of the pancreas have this pancreatic cancerous behavior, whereas when the KRAS mutations are off after a certain time, they see that uninvolved pancreas are high. And similar results they observed for when they use anti-CD4 plus T cells. But for the anti-CD8 T cells, they see that more area of the more percentage of this pancreas are covered with this pancreatic cancers or PDAC behaviors. And once they identified, so far they identified that it's the CD8 plus T cells that plays an important role and interact with the FAS receptors. So they wanted to check what kind of CD8 plus cells interacts with these FAS receptors. So they did the Disney plot of the RNA-seq data, and among that, when they did the meta cluster of these T cells, they are able to cluster them in the 5 different categories. And when they compare this transcript data, or when they overlap this meta cluster with transcript data, they are able to find that most of these FAS receptors are in line with this meta cluster 4. That can be seen in the panel I, and also that can be seen in the panel J, where we can see that the CD8 plus T cells are more predominantly expressed in the meta cluster 4, and FASL is also more predominantly expressed in the meta cluster 4. There are some fast receptors. There are some CD8 plus T cells in the meta cluster 0 also, but there is no fast receptors. Fast receptors are in aligning with the CD8 plus T cells. CD8 plus T cells that can be seen. That can be seen from the panel I and panel J. And once they identified that it's the CD8 plus T cells that interacts with the that has this FAS ligand that interacts with the FAS receptors on the pancreatic tumor cells. Next, they wanted to check whether this KRAS will cause epigenetic mutations that will lead to the decrease in the FAS receptors. So they, in order to check that, they check the FAS methylation in different conditions, FAS promoter methylations, and what they have found out, when the KRAS mutations are on, the methylations are high. That leads to the suppression of these FAS receptors. But when the KRAS mutations are off, these methylations of this FAS promoter region goes down. That can be seen here. And similarly, they also check these FAS positive cells. FAS positive cells, when they inhibit this DNA methylations, and they can see that when they use the DNA methylations, the FAS positive cells goes up even when the KRAS mutations are on. But there is a upper limit, that is when the KRAS mutations are off and they use the DNA methylations inhibitor, they do not see further increase, because FAS receptors reach to its saturation stage in that conditions. But when the KRAS mutations again turn on, then they see that FAS receptors again goes down. So this indicates that KRAS has a inverse role with the FAS receptors. And thereafter they check some of the naturally occurring methylase in DNA methylase transferase, and what they find out is that DNA methylase transferase such as DMT1 and S100Z increases when there is KRAS mutations are on. That can be seen here, that when the KRAS mutations are off, this methylase transferase goes down, or this day 3 off and 5 day off. But when they again turned on, the DLA methylase transfer that help in the methylations of the DNA goes up. And then they check, and these things are quantified, in the panel N, where we can see that when the KRAS mutations are off, this DNA methylase, different kind of DNA methylase goes down. That can be seen here for the DMT1, EZH2, H3K27, Me3, and H. So these threes, and for this H3K27AC, this conditions occurs when the KRAS mutations are off, and that will cause the methylation of this H3K27 Me3, and this acetylations of that will lead to the increase in the expression of the FAS receptors. That can be seen here. With this level increases with the KRAS mutations are off, and at the 5th day after the KRAS mutations are off, the increase is statistically significant. So in the panel O, they shows their hypothesis what could be happening. So when the KRAS mutations are on, this EZH2 and DNMT1, and also this H327Me3 prevents the transcription of these FAS receptors. However, when the KRAS mutations are off, then DNA methylase transferase expression goes down, and also the acetylation of this H327 HEK27 acetylation occurs, and that will helps in the transcription of this FAS receptors, and that will cause overexpression of the FAS on the pancreatic cancer cells. And next, they wanted to check the in vivo immunostaining of these CK19 FAS receptors and CC3, which is the signature for the apoptotic factors, and they can see that when the KRAS mutations are off, this combined positive cells increases, indicates that apoptotics of these tumor cells will start to occur in the in apoptotic of the tumor cell starts to occur where they have the FAS expressions, and these positive cells are further quantified in the panel Q, where we can see that FAS plus CC3 plus CK19 plus cells increases when the KRAS mutations are off, and this increase is statistically significant. So together in a gist, the experimental result presented in this figure points out that this KRAS suppress this FAS expression epigenetically and has a negative role or inverse role. KRAS has inverse role with FAS expressions, and the FAS can interact with the CD8 plus T cells, more precisely CD8a T cells, and that cause the triggering of apoptosis by the CD8 plus FASL ligand that are present on the CD8 plus T cells after its interactions with this FAS receptors that are expressed on the pancreatic tumor cells. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.